my project is really at the interface of chemoproteomics and genetics. Chemoproteomics is the specialty of the Bacchus lab, and human genetics the specialty of the Arboleta lab. So the aims of my dissertation are: one, what can genetics do for prioritizing chemoproteomic detected residues? And the second big aim is: what can chemoproteomic detected residues do for prioritizing missense variants with pathogenic potential? So what are residues though? So residues or amino acids are the building blocks of proteins. There's 20 naturally occurring amino acids, and here shows they're colored by chemical properties. Some of them are essentials, shown by the dashed line, so you have to get them from your diet, and others are made by the body. So these amino acids have different abundances in human proteins, and you can see here some of the chemical properties are more abundant, like aliphatic, for example, the red bars, compared to the sulfur-containing amino acids shown in yellow, cysteine and methionine here. But amino acids and their abundance is actually determined by the sequences in our genes. So this shows the central dogma where you have nucleotides that are essentially used to encode the amino acids that make up proteins. But one gene does not equal one protein, actually, and that's because of alternative splicing. You can have different arrangements of the exons, and how those come together will mean different proteins that have slightly different structure and slightly different functional capacities. So this really ends up being a huge vastness of the human proteome compared to the genome. So most proteins in a human cell actually lack functional characterization and structural characterization because of this vastness. And the dream of the Bacchus lab is to be able to take any protein in a given cell type and ask two key questions. What is that protein's function, and is this function manipulated by chemical binders, or can you manipulate the protein's function? So chemoproteomics is actually this platform that you can test the reactivity of specific amino acid types, and you can profile these amino acids in terms of their ligandability to small molecule probes or their reactivity. And this here shows a cysteine residue zoomed in, and it's reacting with one of these two types of chemical probes. So other amino acids can be detected by chemical probes, and for the purposes of my study, we're going to look at lysine and tyrosine as well. So chemoproteomics is a really important technology and platform that's facilitated the discovery of lead compounds to treat human diseases. And here's some examples of blockbuster cancer drugs. And really the question of my project is: how can I support the identification of other therapeutic drug target sites? So the challenge with working with chemoproteomics data and these probes that can selectively label a specific type of amino acid is that there's thousands of residues detected in a single experiment. And so given this protein you see here, in the yellow is sites of cysteine residues, and on average there's about 13 cysteines per protein. So residues that cannot be labeled by a chemical probe are buried, and that's shown here by the black color on the structure. But there's other reasons why a chemical probe can't label or detect a residue. For example, post-translational modification, redox oxidation. But even though you can't detect all residues, of the ones that you can detect, it's still a challenge to figure out which ones you should target for maybe to inhibit the protein function, or restore or enhance function, or confer novel function. So identifying labeled residue sites that are functionally important is the first aim of my project. And here shows G6PD was a protein we studied. As you can see, there's multiple colored sites that have been detected by a chemical probe on the structure, and this is highlighting the active site lysine residues. And you can see that even with one protein, there's still a question of which which amino acid site should be detected by a should be targeted selectively by a chemical probe. So what can genetics do to help this problem of prioritizing important chemoproteomic detected amino acids? Well, it reminded me of a central challenge in human genetics, which is interpreting the disease relevance of missense mutations. So a missense is a single amino acid substitution in a protein. And leveraging the unique relationship between compound labeling and missense mutations can help inform which detected residue should be prioritized. And here shows a protein in the side chain is changed because of a compound labeling event. And then on this side, you see a missense mutation from a cysteine to a tryptophan. And both of these modifications or changes can have the following functional consequences, such as inhibiting the protein's function, conferring neomorphic activity, altering its interactions with other proteins, and so on. So the goal really here is to apply genetic variant scores that are made to predict the consequences of, or the pathogenicity of missense variants to chemoproteomic detected residue sites. So here you have a protein sequence with lysine and cysteine detected sites, and the idea is to score these positions using deleteriousness scores from genetics. So how do you essentially do this? Well, you have to start off with doing residue level mapping, which is looking at a protein and the detected residues on that protein. And there's a question of which isoform of the protein sequence should you use. But once you get past this level, the next thing you need to do is DNA level mapping, and essentially converting the amino acid positions to genomic coordinates. And there's a question at this level of which genome assembly should you use. There's two available, GRCH37 and GRCH38. And then the final step of which functional, or annotating these detected residues with functional annotations from genetics to interpret the impact. And you can see some examples of annotation sources we used for the first part of the project here. So residue level mapping to DNA is like doing laundry, where you might have 8 socks at the beginning, and after the laundry, you have 5.4 socks. So my question with mapping was really at the beginning, what caused sock loss or residue loss, and can this loss be prevented? So part of the reason there's data loss is because the databases that are used for sequence information like NCBI, CCDS, GenCode, Ensemble have frequent updates and they're asynchronous. And you can see data sets that were needed for our project over here, some overlap certain versions. For example, the CAD score we used is an Ensemble, based on Ensemble version 92 and GenCode 28. But then there's other scores we were interested in using, like Dan and Fathom, based on Ensemble version 94. So it basically comes down to when you lose socks or when you lose residues, it's partly related to these asynchronous data cycles, but also a reliance on cross-references, which is how you go from one database to another. These cross-references can be outdated, and not having confidence in your reference between sequences is a problem and can lead to mismapping errors. So we wanted to, understanding this problem, actually quantify what proteins are at risk of mismapping. So we identified sequences that were most vulnerable to residue level mapping. You can see here by mapping, I mean essentially taking a uniprot protein sequence and checking that that sequence, which is mapped with the identifier for the uniprot matches is cross-referenced to the Ensemble protein identifier, and just checking to see if the two sequences that are supposed to be or assumed to be identical actually are. And to answer this question, we use the Hamming distance, and just to give you an example of what this distance would be for changing my name, this is a distance of 2. So when we checked if these two proteins that are cross-referenced are essentially identical across multiple versions of Ensemble, you see that in the 85 release of Ensemble, there's large differences between a cross-reference protein to the uniprot protein, and that's shown by the darker blue color. And then looking at the other releases, you can see how this changes. So these were some genes that were most vulnerable to residue level mismapping, and it's important to be aware of how this can affect your data set for research you're interested in doing. So our solution, discussed further in the paper, is really about creating a validated cross-references to enable high fidelity residue level mapping. So now let's get to the problem that we've solved mapping. So connecting predictions of DNA variant pathogenicity to chemoproteomic detected sites. And here you have, on this protein, let's say you have cysteine position 5. How can I score this with missense pathogenicity? So the first step is in silico mutagenesis of this cysteine's codon. And this codon for the cysteine is TGT. And you can change every position to every other possible nucleotide, and you get these codons. Then what are the possible amino acid gains? Well, these are shown here, and we did not include the stop codon or nonsense mutation and the silent mutation, so just changing from cysteine to cysteine. So we're only focused on missense. And then we score these missense. And taking the max or the mean, we connect that back to the original cysteine position. And the score that the results in the upcoming slides show is related to the CAD score. So, chemoproteomic detected residues were found to complement missense pathogenicity predictions to identify functional amino acids. On the x-axis is the protein positions of caspase 8, and the y-axis shows the adapted CAD score that we just reviewed. Now, if you look at the pathogenicity scores across the sequence, there are 2 detected residue sites in this protein, cysteine 360 and cysteine 409. 409 was just below the threshold that we used from CAD that defines essentially anything above that score of 25 is pathogenic, if lost. Cysteine 360 was a known site of a pathogenic mutation in uniprot, and it's in the active site of the enzyme. Cysteine 409 had 0 functional annotations at the time. We found that loss of function mutations in this enzyme are associated with an autoimmune disorder. And when cysteine 409 was validated in the wet lab experiments, we saw that mutating cysteine 409 did kill enzymatic activity. So this is caspase 8, and it's colored by the CAD score. This is showing an inhibitor that's known to bind the active site of the protein. And you can see that the active site is really high CAD score, which is, expected. So let's look at those 2 residues. So cysteine 360 and the detected cysteine 409 are highlighted in red, and we check the distance of these 2 sites to the known inhibitor. You can see that the cysteine 360 is closer to the inhibitor site, and cysteine 409 might have been missed if this complementary method wasn't used to prioritize it. So this proves that integration of genetic scores is a powerful tool to identify functionally important detected residues relevant to human disease. You can check out more results like this and more discussion on mapping and tips for mapping in our published work. And there's also a companion Shiny app to view the results. Okay, so second aim of the project is what can chemoproteomics do for prioritizing disease-relevant rare missense variants? So there's a high burden of uncertainty in rare missense classification. There's, in everyone in this room, about 500 rare missense variants, and 1 to 2 are de novo, so that means never before seen in the population. So this is the most abundant type of protein-altering variant, and most of them are classified as variants of uncertain significance. And you can see this is a curation of clinically relevant variants, and it's called ClinVar. And if you look at the missense breakdown of classification, 93% of missense in this database are classified as variants of uncertain significance. So unique solutions are required to identify the needle in the haystack, or the pathogenic variant amongst the sea of benign. So Omen, most of these missense variants that are classified as VUS are in Mendelian genes, and Omen is a resource of phenotypes for rare disorders and the genes. There's about 8,498 as of June 2021 rare monogenic disorders described in this database. 71% of these disorders are caused by mutations in 4,212 genes. The remaining 29% of these disorders have unknown molecular bases. And focusing on this 4,212 number, that's only 20% of genes currently in the human genome implicated in rare disorders. And I think there's many more, and so more research is needed. Another reason this aim focuses on rare diseases besides the high VUS burden is because there's a significant unmet need for therapies available for rare disease patients who have serious and life-threatening disorders. The FDA-approved medications only account for 5% of rare disorders, so that's 95% lack targeted therapies. So we wanted to see how the overlap of chemoproteomic detected proteins compared to FDA-approved drug target proteins and our set of monogenic disorder genes. And you can see that there is significant overlap between the detected proteins and Omen genes, which I'm abbreviating as Omen, for monogenic disorder associated genes. And a lot of this overlap does not, or excludes the FDA drug target list. Another important thing we wanted to look at when prioritizing how detected proteins could be applied to identifying rare variants with pathogenic potential is constraint. And constrained regions in the genome are essentially regions that you see less variants than you would expect. So looking at this gene structure, these are the exons in the gene, and all these black dots represent variants identified in the population, so protein altering mutations. You can see some exons have more of these protein altering mutations that were in healthy individuals, and some exons are completely devoid of these mutations. So you would call this exon minimally constrained to mutation because mutation is allowed there and it seems to be fine. It doesn't affect the person's health. And then you don't see any mutations in the population occurring in these regions. So you would consider this a highly constrained exon. So a whole gene can be classified as well as missense constrained if you look into Nomad's metrics for gene level constraint, and they have a certain threshold of 0.35 for their missense constraint metric. So we wanted to know how many detected proteins have a depletion of missense variants, and we found that 83% of the missense constrained genes in humans are actually also detected proteins from our chemoproteomic experiments. And as a control, we wanted to compare to loss of function tolerant genes. So these would be genes like the olfactory receptors, where you see common loss of function mutations, and that's allowed for the healthy population. When we looked at the 95, this overlap here, the 95 proteins that have been detected in chemoproteomic experiments but are also missense constrained, you can see that in this protein-protein interaction network, they're highly connected. There's enrichment for disease associations, including intellectual disability and developmental disorders. And most interestingly, 56 of these missense-constrained chemoproteomic detected proteins have 0 rare disorder connections. So these might be interesting genes and proteins to follow up on to identify novel disease gene associations or connections. So to better understand what features distinguish a pathogenic missense and a benign or neutral missense in this particular set of genes, the Mendelian genes, we wanted to understand the abundance of both the codons and the amino acids that would influence the spectrum of missense mutations. So omen genes contain significantly less cysteine compared to all other proteins. This plot shows essentially the normalized difference between the mean abundance of the omen genes versus all other proteins. And you can see by the chemical properties colors that cysteine, again, is very low abundant, especially for this particular protein set. We also looked at not only the amino acid abundance, but the codon abundance in these genes, and saw, interestingly, if you look here, arginine, it seems like the arginine amino acid is depleted in these genes, but looking at the codon level, not all of arginine's codons, there's 6 of them, are actually depleted. There's an enrichment of the codons that are particularly mutable, which is the CpG containing codons of this amino acid. So why does that matter? Well, the abundance of codons, like we saw with the central dogma, is going to influence what missense are possible in these genes. So the next thing after understanding these abundance features was to look at the enrichment of missense mutations that are known to be pathogenic and known to be benign in omen genes. So here are the results. When you look at pathogenic, when you look at loss by missense and gain by missense, there are essentially enrichment for loss of cysteine, tyrosine, and gain of lysine over here. And I'm pointing out specifically the types of amino acids that are detected by chemical probes. And this loss of cysteine and tyrosine is interesting because that might mean that there are chemical probes that can target these residues, and if they're functionally important, you would see a change in the protein's function. And then for gain, seeing that pathogenic is enriched in this particular set of proteins means that there might be future drugs that could target these gain of lysines selectively. And here's some example of rare disease targeted therapies. For example, the therapy for cystic fibrosis targets a gain of an amino acid. And using this, like pointing out the lysine case here, we hope that future therapies that could be developed from this analysis. Okay, so understanding now the mutational landscape that's specific to the Mendelian genes, we wanted to look at the distribution of missense around the detected residues. And we looked at both the 1D windows, so that's just the sequence of the 1D sequence, and looking at either a ±3 range, and that's shown by the gray box here, or a ±6 range, or ±15. And to do this accounting, you can see that in this particular detected residues window, it's shown by the little asterisk here, there are 3 pathogenic missense. We also looked at the missense variants that are in the detected residues local 3D environment. And having all of this data, we wanted to see if we could apply, similar to our first project, our approach to understanding or identifying disease-relevant variants. And we collaborated with the Christoph lab and particularly Blake Wild was kind enough to share his functional validation of fumarate hydratase. This is a tumor suppressor protein. It's part of the TCA cycle. It has many VUS identified in it, and other pathogenic mutations that are known to cause hereditary forms of cancer. So he screened 74 VUS as part of a preprint that is out now, and we wanted just to compare the results of his functional validation of these FH VUS to detected positions of amino acids in our data set. So fumarate hydratase has 16 amino acid positions that have been previously detected, and they're shown here. So there's 2 cysteine positions, 13 lysine, and 1 tyrosine. So we use the local 1D and 3D missense accounting data to look specifically at these 16 chemoproteomic detected amino acid sites in FH. And when we clustered by correlation, we saw that cysteine 333, cysteine 434, and lysine 333 were at the top. And all 3 of these detected sites have a CAD score above our deleterious threshold of 25. So FH, is important to note, is actually a protein that is functional when multiple copies of itself come assemble together. And those are called multimeric proteins. It's just one of biology's ways of having the protein diversity that we see with not as many genes needed to have that diversity. And essentially this is a monomer pepsin, and FH is a homotetramer, which means it's got 4 copies of itself coming together in a particular orientation. And one way that missense can be pathogenic is by disrupting the protein's ability to form into this multimeric structure. So Blake's data, he shared with us the multimerization data he had, and we saw that there were VUS overlapping 3 detected residue sites. That's cysteine 333, cysteine 434, and tyrosine 465 shown on the x-axis. And cysteine 333 was prioritized by our approach, and it actually had the greatest disruption of FH multimerization seen. So this was another example of how chemoproteomics can complement looking at rare variants and interpreting the possible pathogenicity of these sites. So bridge conclusions. Rigorous and reproducible mapping standards can reduce residue data loss and enable high quality annotations. Genetic pathogenicity predictions in our first project did support the prioritization of functional chemoproteomic detected amino acids, and we saw that with caspase-8. And lastly, chemoproteomic detected amino acids and their associated missense variant environments can support identification of VUS with pathogenic potential, like we see with fumarate hydratase. So I've had the privilege to work on other publications during my PhD, including a study on Cat6b from the Arboleta lab, and also a project from the Bacchus lab, CysTV, and a review from the Ruby lab on sex differences in obesity, lipid metabolism, and inflammation. I'd like to acknowledge everyone in both labs, my committee for getting me to this point in my career, and especially my mentors. I think it was definitely a high risk for someone like me to take on this type of project that's very computational. For example, this is a drawing from the first meeting I had with Carrie and Val. And it's really clear to me now I had no idea what I was doing, and I was asking to do an entirely computational project, and I did not know how to code in R and Python. So I'm incredibly thankful that they let me follow my passion, and yeah, just thank you for believing in me. I also want to dedicate my thesis and dissertation work to my late grandfathers, Mario and Charles, and to my best friend, Jose Omar Castellon. And then these are just people I love and have made this a very memorable experience for me. So my mom, my Nana's on Zoom, my dad, my sister, and my Austin family, my friend Sophia who came from out of state here, my Nana listening to my project, thank you, and other friends. So yeah, thank you so much, and that's all.